Howdy, my name is Carly Skelton, and today I will be presenting on how Apis mellifera resistance has been influenced by destructor mites. So a little background is that Apis mellifera, aka the western honeybee, is of huge economical, agricultural, and ecological importance worldwide, and it's part of the order Hymenoptera, along with wasps, ants, and bees, and is considered the most beneficial order to mankind, mainly because of their evolutionary adaptation to pollinate crops and plants, and some have also evolved into parasitism and predation of pest insects. A little background on the mite is that it's called destructor, and they're little brown-red external parasites to bees. They have caused millions of hives worldwide to die and are one of the largest pathogens to Apis mellifera. It is also able to feed on the bees, weakening them, malforming them, and inevitably crippling a lot of hives. They are also huge vectors or transmitters of diseases between the bees, such as deformed wing virus, which we have seen spread. So a little background on mites. The problem began in the 1980s, and the reproduction occurs in the brood. And these infestations can build up in 3 to 5 years, and symptoms can't be seen until a large infestation has occurred. But the brood is basically bee eggs that are found in the brood nest of the hive, and they're part of the holding cell where the mite may lay 2 to 5 eggs, and it's usually after the brood cell has been capped. So these mites then go on to parasitize both the baby or the brood, middle age, and adult bees. So it's a parasitic interaction, a negative interaction where the bees are hosts to these predator mites, which parasitize them. So the accumulated effects on the entire colony can cause reduced colony populations, queen supersedure, or colony collapse at its worst. So it can weaken the immune system of bees in terms of the individual as well as in terms of the entire colony. So their physical appearance are typically that the males are lighter in color and smaller than the large, dark females. And these mites will readily infect European honeybees, even though they originally infected Asian honeybees. Along with that, what I mentioned earlier, the mites have formed a parallel relationship with the deformed wing virus, and both of them are increasing in numbers within bee populations in current years. So as we can see, there are different effects on the colony versus the individuals. So if we look at the individuals, we'll see that the mite will cause them to reduce in weight, impair their flight abilities, cripple and cause crawling of the bees, reduce their lifespan, reduce their abilities to forage, and cause a scattered brood. In terms of the colony, it causes a parasitic mite syndrome, which can lead to irregular brood patterns, abnormal sunken brood cappings, slumped larvae in their cells, reduction in populations, supersedure of queen bees, and then inevitably colony collapse or death. So why do organisms build resistance? Well, if you look, a pathogen is typically an organism like a bacteria, virus, single multicellular eukaryotic organism that has the ability to cause disease onto another organism. The degree of the disease is measured by virulence and virulence of the pathogen. So a couple examples of this are cotton wilt fungus, which is a fungus, and it typically causes cotton plants to die, but it works with bacteria, and it's a pathogen onto cotton plants. Zika virus works through mosquitoes and is a pathogen to humans and animals. A parasite, on the other hand, is an animal or plant which can obtain nutrients from another organism either by living on or inside the host organism. And this causes the host to slowly die out or to perish immediately, depending on the parasite. But a couple examples of this would be the veromite, as we've been talking about, and then Naegleria fowleri, or brain-eating amoeba bacteria, which you can find in lakes in the summertime when it's really hot. But those are just a couple examples of parasites so that you can see the difference between a pathogen and a parasite, because a parasite is a bit more invasive and typically leaches quite a bit of nutrients from its host. But in terms of that, we want to look at resistance. And resistance is an attribute that quantifies how much an organism is able to fight against a parasite or a pathogen. So this attribute can be seen in antibiotic resistance, where bacteria have developed the ability to fight against the drugs which are intended to kill them. And a graph looking at that is resistance example using bacteria. So as we can see in the graph, you have non-resistant bacteria, which are then exposed, and they then multiply, and eventually they die and the person is healthy again. But as soon as you get drug-resistant bacteria, these bacteria now have a resistance gene within their genome, which can then multiply, divide, and then this bacteria continues to spread, causing the person to remain sick because the bacteria is not going to be affected by the same antibiotics that once caused its decline. And that is because over time, just as we see in nature, there's variation and then there is adaptation. And this causes our evolutionary adaptation to, drive evolution, drive the change of our species. And resistance, especially to things that harm us or harm these bacteria, is going to be resisted. So how has Apis mellifera resistance varied as the verodestructor parasitism issue has increased? That was my hypothesis and question, just the kind of guidelines that I was following whenever I did my research. But resistance in Apis mellifera. So resistance, once again, is the ability for a population to fight off a parasite in our case. So in the 1950s, the mites jumped from the Asian honeybees that I mentioned earlier, Apis carana, to their present host, Apis mellifera, the European honeybee, along with the rise in deformed wing virus, also known as DWV worldwide. The mites and deformed wing virus is a worldwide issue affecting every country except Australia and a few small isolated islands that have never been exposed to these issues. But we saw resistance formed in the presence of deformed wing virus in the absence of treatments. The bee populations initially suffered colony losses, but then formed resilience where there was an absence of acaricide exposure. So acaricides are typically used in more modern countries. They're fighting against ticks and mites. And in the absence of those treatments, the honeybees were then able to be exposed to the mites, maybe suffer a couple of losses initially, but then form a resistance and then go on to feed that generational resistance. So as we can see in the graph, if there were more recappings, you can see that there was more mite detection. So the bees were more aware that the mites were in their cappings and were able to clean them out. So the resistant ones were then able to clean out their cappings a little bit better than the ones that were susceptible. And yeah, I just wanted to show the susceptibility versus the resistance. So as you can see in graph G, there was an increase amount of DWV, or deformed wing virus, load in the susceptible population, but the resistant one had a smaller mean of deviation. So there was less, I guess, loss. So, another thing I wanted to bring up is interspecific differences. So, something between the Africanized bees and the European honeybees, because in Africanized honeybees, they were resistant to the Vero mites. This was interesting because the first mite-resistant bees were found in Africa and in Southern America in the Africanized honeybee, also known as AHB. So I looked into the predation and tropical influence, seeing that there were more swarming events since there are higher temperatures and more predators year round. So that keeps both the bee population on alert for a new hive, but also absconding any type of nest that isn't fitting their needs anymore. So, also a factor that went into this resistance was a lack of acaricide, chemicals that kill ticks and mites. Use allowed resistance to occur in the feral populations because of the initial affection, loss, and then the resilience of these colonies. A couple of practices that we saw in the resistant populations included brood removal, where the Vero-infested pupae were removed from the nest and there was no chance for them to infect further. Number two was recapping, where the holes that are created allow direct access to the pupa and then are resealed. So they're checked, resealed whenever they are checked to be clean. And then three, mite infertility, where female mites are unable to produce viable mated female offspring. So in that case, they wouldn't have offspring that can continue their bloodline. So in conclusion, Apis mellifera resistance has been positively influenced by Vera destructor parasite. And Vera mite has caused the European honeybee to develop resistance within the adaptation of multiple practices which keep the mite issue low but is mainly utilized by the Africanized honeybee. These practices are critical in keeping low infection numbers in the hive. Veroparasitism, arguably coupled with virus infection, result in significantly higher transcript abundances for the antimicrobial peptides abacin, hymenoptecin, and defense 1, Bidla 2012. And in conclusion, the biggest point I would like to make is that Vero mites with deformed wing virus and an absence of acaricides will lead to resistance in the Apis mellifera populations, such as in the Africanized honeybee populations, which have found increased resistance to the mites after being exposed initially. So significance of this research is that coupled with the deformed wing virus, studying the two largest pests for Apis mellifera populations worldwide is very significant. It'll enhance the survival of future honeybee generations, just finding more information on this pest. Three, we could understand why the honeybee has had to form resistance and how it will continue to change in the future. Four, it will ensure the mite issue cannot further damage current colony populations once the solution has been discovered. And altogether, it'll encourage healthy apiaries with prevention practices. So here are a couple of my sources cited, and that's all I have. Thank you, and I really enjoyed this class.